our next speaker, hi, there you are. If you want to get your slide set up while I introduce you, that's fine. Okay, you're going for it. So I'm happy to introduce Dr. Akasha Faist. Dr. Akasha Faist is an assistant professor in the Department of Animal and Range Sciences at New Mexico State University. Her current efforts are focused on rangeland restoration ecology, where she and her research group work to find ways to overcome barriers to restoration success and improve ecosystem function and biodiversity. Today, Akasha will be presenting on creative approaches to enhance Southwest restoration success. Go ahead and get started, Akasha. Thank you.
Great. Thank you so much. Yeah, something just happened with my screen and you disappeared, so I will pay attention to the time before to make sure I know what the time. Yeah, thank you so much. Yeah, I'm going to be talking today about, as Melanie said, enhancing restoration success using kind of different creative ways. So I'm an assistant professor here, and our research group, we spend a fair amount of time thinking about, well, what do we think about, I guess is the question. We think about what do we need to do to enhance restoration success? This is a question we almost start every project with, every problem. What is the need? What is the need from an ecological restoration standpoint? We often work in rangelands and in more dry systems, but we also work across systems as well, asking this question. And then we say, okay, so what is the need? Let's have our evaluation of tools. And ideally, what this all comes down to is we need to identify the need, the evaluation of tools, and really we want to identify novel and creative actions to overcome barriers to ecological restoration success. We know they're there. Well, what's the need? What are the tools that we can use to create, to overcome those barriers? And we take an approach both in the ecological restoration, where the actions, what are the actions taken? Is in this case, it's using fire as a restoration tool, using other actions and restoration tools, and then using the ecological knowledge and assessing those. So thinking about thinking about over time, how do we improve ecosystem function, getting our degraded state to a more reference ecosystem. So how do our actions create outcomes? And we also think about this in a cooking, baking perspective, where so the actions as a restoration ecologist, as a practitioner, as many of us know, the actions that we take can actually determine those outcomes of the restoration project. Do we add more flour? Do we add granulated sugar? All these different interactions, these actions that we're taking can create a different trajectory, and we could have a different outcome at the end. So maybe we want to be a little creative with our actions and see, well, we want to enhance and we want to overcome these barriers and enhance restoration success, kind of following that ecological framework. What actions to take? I mean, this conference and so many others, these are big questions, right? So we try to kind of piece those together and say, well, let's figure out the best course of action. First, identify the need. What are the barriers hindering success? What is that chance of success? Do we have a high chance of success, low chance of success? If we're actually intervening, we have the active restoration. How much intervention is needed? And then we think about that with that, those are going to be interacting with each other. Now, propagule availability is something, again, I'm kind of framing how we approach each topic and each different action that we're taking. And we spend a fair amount of time thinking about, well, are those propagules, are those seeds, in our case, we use vascular seeds a lot, is that propagule availability there? And so how does this all come together in a framework of the seeds? So how much work do we need to put in? What's our chance of success? And so we ask a big question about what seeds are already there. And this in the form of the soil seed bank. So we have a major component of our work is looking at the soil seed bank, and that's the storage by plants. So the soil below the ground, I mean, we talked about them in this and others, where that's storage by plants. So how does that help us with the restoration actions? How is this a potential creative way of thinking about it? Those seed banks are there, the dynamics are there. They provide, as we are familiar with, the history and the future of a system. So they can kind of help us understand not only what is potentially going to be there in the future, but what was once there. And they can be highly diverse. They can actually have a higher diversity than the associated above ground vegetation because they have different space and time considerations. Their storage, right, as we all know, there's storage of these little propagules. I call them, they're packets of infinite potential because they are storing that below ground, and they can actually have a higher diversity than maybe what we're seeing. So we can actually understand the potential of a system that if we want to go restore it. Whether it's invasives or native, knowing what's in the seed bank can help us inform those restoration efforts. So thinking about the soil seed banks can kind of help us inform our restoration efforts. So we have a number of different studies on this. I'm going to just key in on one here and then kind of overviews of a few, but I'm always happy to talk about seed banks, always, always, always. So definitely don't hesitate to reach out to me if you have seed bank questions. So to approach soil seed banks in kind of a restoration framework and how they could potentially be, and how we could think about them in our lab. So Kirsten Romig is a scientist at the Jornada Experimental USDA ARS. And so she's looking at a study where we have across this system, there's these different ecological sites. So there's, and so these ecological sites share these similar abiotic and biotic characteristics. So they're grouped together where the clay soils, and they have a similar plant community above ground. And this has all historically been done above ground. So the clayey sites, the shallow sandy, the loamy, the sandy, these ecological sites have all been kind of an above ground kind of lumped together. So what she did was, here you can see on the graph, there's these different ecological sites. So clay, loamy, gravelly, limestone hills, sand hills, et cetera. And what she found was that the sandy sites actually had more seeds, and this is seeds on the y-axis, it's seeds per meter squared. She actually found that the sandy sites, irrespective of their above ground vegetation, because the shallow sand, sandy, deep sand actually had different vegetation structure above ground, but the sandy sites actually had more seeds. So the seed density in these soils were higher than clay, loamy, and gravelly, which all have very different soil textures and also different vegetation. But so this was really exciting to us, because finding that these sands, so it was almost a texture, and there's nuances that I would love to talk about, but the texture actually kind of improved. The seed had higher seed density, almost more so than the vegetation that could be depositing those seeds. And this also, something that really stood out to us was, so she also looked, and I don't have the data here, but an overview of, she looked across degradation. So within each of these different sandy sites, deep sand, which of these ecological sites, she looked at the degradation across them, and it still was the texture that was kind of guiding the densities of seeds. The diversity we're still looking at, and we're kind of building on that, but the density and the number of seeds actually was driven by the ecological site. And in particular, we were thinking it was the soil texture that had a really strong component. So that's pretty neat, right? So, and we're building on this with future studies. We're actually looking at building seed bank suitability models so that we can use that in our restoration to say these soil properties have the best potential for seeds. And so then we could use those to then influence our restoration actions. So it's kind of fun. So what does this mean for restoration? So seed banks and restoration. So the seed bank composition, the soil, and I'm referring to the soil seed bank composition. There's such phenomenal work about collecting seeds, but we're saying the soil seed bank composition, similarity to the knowing how similar or different it is to above ground vegetation can actually guide our restoration efforts. If there's a mismatch with the above ground vegetation and the below ground seed bank, well, what is that? What is happening there that can actually guide our restoration efforts? And a big thing that we spend a lot of time thinking about, we can minimize the duplication of efforts as well. So one thing across many studies that we've found, many studies, that Spirobolus, the genus Spirobolus, they're tiny small seeds, right? They make a ton of seeds. This is a perennial grass. They are everywhere. They're across multiple soil textures, they're across multiple different deserts, they are everywhere. Well, what does that mean if we want to actively seed on using restoration? Let's maybe we want to use species that are not already in the seed bank, because we can learn a lot. If they're in the seed bank and not germinating, they're maybe telling us something, that the conditions aren't right for them. Or if they're already in the seed bank and we want to actively complement and not duplicate, our active seeding can actively place those seeds and actually not duplicate efforts, excuse me. And we can also maximize competitive advantages. So we often think of seed banks as being very highly invasive. And this is, in some cases, is absolutely true, but in some cases it's not. But knowing if there are invasive species present, we can add seeds in our restoration action that would actually outcompete with those in the seed bank, but we would have to know what those are first. So thinking about that, thinking about seed banks critically, what seeds are already there. Another thing we wanted to think about too is so there's the seeds that kind of more permanent storage. Well, will the seeds get there without us, is another question that we want to ask. And so this is here outside in southern New Mexico here. So the question of woody plant encroachment is a big concern we have, where where we have this grassland to shrubland transition often. And this is a study done in the Jornada experimental range, where you can see visually, actually, here on the bottom left, you have kind of a more continuous, darker gradient. Those are grasslands. And then in the top right, you have shrubs, and this is mesquite encroached shrubs, and you actually cross a threshold and you move over to a shrubland where you have a lot more open space and a lot more interspace, and there's a lot more potential for sediment movement across systems where you have these islands, as opposed to a more continuous vegetation cover as you would in these grasses. And here's just a close-up showing that. So you can see these there's streetways or these pathways where if you get windstorms, you have kind of the momentum of sediment and seed can move. Okay, so here you can have, so what we did is there's these connections, these pathways that are connecting. So you can have these connectivity modifiers. So we can, as a restoration action, we can actually put in connectivity modifiers, and these have been used across a number of different systems. RestoreNet uses them, and Dryland Network uses them, and others have used them to kind of modify the connectivity. So I had a graduate student, Tyler Turk, who asked this. She said, well, if you have more connectivity when you have more shrubs on the landscape, how does that matter with seed movement, and what would that matter for our restoration actions? So what we did was, so she and this is out at the Jornada experimental range in southern New Mexico, we had a mesquite encroachment gradient. So here she had, so this is Tyler's study design, where she had these big plots put out that had subsets within them across a shrub to grassland gradient. And she did find, and these are each the different blocks at the broad scale, that the grass had less bare ground than the shrubs, and there were increased connectivity. So there was more movement across when you had more shrubs. So there was more bare ground and more movement, those pathways, more potential for sediment to move through. So she wanted to say, well, okay, so this is happening. We talk about resource distribution. Well, what's potentially happening with those seeds? We know that there's the seed bank and the soil seed bank. Well, what's happening in the above, or in the transient and the sediment seed bank? So she put in these ConMods across this gradient in the summer and late fall, so our kind of monsoon season when we have most of our seeds dropping. And she collected the sediment that was deposited in these ConMods during that time frame. And that was the sediment plus the seeds plus the litter plus everything. She collected all of that in there. And so what she did was, and this is the same as in a soil seed bank study, she did what we call an emergence trial. So where you take this sediment and when we do other seed bank studies, we take soil samples and we put them in a greenhouse and we give them really optimal conditions, and we try to release the seeds from the seed bank, giving them the best possible conditions, whether that's kind of like gibberellic acid as a hormonal treatment to break dormancy, water, dry down, wetting. So the greenhouse trials are very often, we call them emergence trials, how we release the soil seed bank across seed bank studies. And she did that same idea here, where she took the sediment from that was moved, and this was just within this late summer to fall movement, and she collected the sediment and seeds, and then she did that emergence trial with that sediment that had been moving around. So she was isolating, well, what was moving around out in the landscape. And so she did that emergence trial. It takes a while. We have to make sure we get all those seeds expressed to germinate and break dormancy. But what she found was really exciting to me. I was pretty floored by how many seeds were actually moving. We know there's a lot of seeds moving. She found in our dryland system, so she had these 64 ConMods, this network across this gradient. 62 of the 64 ConMods caught viable seeds, which is cool. 27 genera, so it wasn't just one species moving around. There were 27 genera moving around, but then how many seeds? So there was a mean of 250 seeds per meter squared. Obviously, this is the mean. They were highly variable there, but there was a mean of 251 seeds moving across these different landscapes. So there's highly diverse subset of seeds moving across the landscape, and at a much higher density and diversity than we had originally thought. We knew they were out there, but how much seed movement is actually occurring in these connected pathways was pretty exciting. And so with that, so we said, well, it's got to be a 1-to-1. If you have more sediment, we hypothesized it's got to be a 1-to-1. If we have more sediment, there's probably going to be more seed counts. So what we did here is, so she did the volume and the grams, and there was a 1-to-1. So she actually here did the weight, apologies. So she had sediment captured on the x-axis and then seed count on the y here. So generally, the more sediment that was moving around, the more seeds you had, which is kind of intuitive, but still thinking about it, if you're increasing your connectivity, you could actually lose seeds from a system if there's so much sediment moving around. But we also wanted to look at, well, what did that matter about the seed size? Who was moving around? How are these being distributed? So we found there wasn't this makes sense, there wasn't as many large seeds. But there still were out there. There were still large seeds that did move kind of, we were thinking, and maybe at like a less, not as far, in the medium seeds, but these small seeds actually had a much stronger relationship. So the sediment that was moving those small seeds, which is kind of neat. And to be said, and to point out that actually it is kind of neat that there was a lot of spirobolus. And so we know from all of our other studies how much of this tiny seeded drop seed was in the soil seed bank, and kind of the more permanent soil seed bank that's kind of maintaining, but it's also the soil that is moving around. And that might actually explain why we see it in so many locations, because it's a highly mobile, those small seeds are moving with the sediment at a pretty high proportion. So they're kind of making up a huge amount of the more persistent and transient seed banks out there, which again, thinking about going back to restoration actions. So if we know who's moving around, our large seeds in this case were mesquite. So if we know there's still those mesquite are moving around and those are seeds we don't want, well, what's the management actions we can take? Or if we know it's other spirobolus or others, well, can we seed these ConMods? And there's been some great work in seeding ConMods. Can we seed these ConMods to then facilitate not seeds that we're going to get there to the ConMods anyways, but seeds that would not get there and that we want and are more desirable. So thinking about that from not only like the seed banking and the dormancy and dormancy break, but the movement, and thinking about our actions in that respect. So again, just thinking about that from what's on the landscape, what are those seeds moving or maintaining as storage? So maybe the seeds are already there. Maybe they'll get there without us. But I think, and as this conference has is wonderful at, we often need to add seeds, right? That doesn't mean that, just the idea of thinking about, are the seeds already there and what do we need to get there, are a really great tool that we like to use, and many others, is what is the tool to then actively seed, because we often do need to add seeds. Maybe we know we have a bunch, we've done seed bank studies, or we know we have a bunch of invasives. Well, do we need to add seeds? Maybe we have a really depleted seed bank. So what are some mechanisms we can do for our restoration? So this is the native plant seeding. Thinking, again, this is the perfect conference for this, many different great ideas, creative seed mixes, thinking about how to, thinking of the community ecology and how are they interacting. We spend some fair amount of time kind of discussing that and thinking about, well, how are they then going to interact with their environment? So the application techniques, I'm going to talk a little bit about that one. And then coupled with habitat modification. So you have the biotic and the abiotic interacting. How do we interact? How do we kind of facilitate that to enhance restoration success? So again, what actions do we want to take if we're going to actively seed on the landscape? So what actions to take? And this gets down to that resource accessibility. So propagule availability, maybe there's less intervention needed because it's a propagule availability, but the habitat of the soil foundation is fine. Maybe it's a little bit more difficult to achieve that intervention, that chance of success. Not impossible, but a little bit different. So here in dryland systems, we're constantly trying to figure out how to overcome water limitations. Classic question we always ask. It isn't the end all be all of the different barriers we are overcoming, but it for sure is a very important question that we all ask many a times. So for the last bit here, I'm going to talk about a study led in our lab by Savannah, who we looked at testing creative soil restoration actions to enhance soil moisture and improve native plant restoration success. So this was done right next to the Jornada Experimental Range. This was done in the Chihuahuan Desert Rangeland Research Center. This is right next door. So same system, very dry system. And what we experience in this system, again, kind of like with Tyler's, that summer where we have the most seeds, we really have these monsoon-driven systems where you get big pulses of rain and then prolonged periods of drought, pulses of rain. And so what we thought about, and we tried to think, well, what could we do, is we thought about using these hydrogels. So using something that can actually capture soil water, and then when you get the big pulse, something that captures soil water, that then when you have these prolonged truncated times between water, that it can then have a slow release of water. So we were thinking, let's do an experimental design and test this. So here, what Savannah did was there are 2 planting treatments. We said, okay, so seeding. So we used 3 species, Atriplex canescens, Elymus elymoides, and Macranthera. We tried to use a grass, shrub, and forb. And so these were seeded at 400 seeds per meter squared. And then another treatment was outplanted seedlings. So this would be six seedlings per meter squared. To see maybe these hydrogels, maybe with germination, it could enhance it, or if the seedling is already there, they could capitalize on that quicker. And then we wanted to say, well, this is, if there's water, we have dry systems. We also had a watering treatment. So we watered, which simulated an average monsoon event. And then we had the ambient, so only received ambient precipitation. And this is just to kind of visually show you the different what hydrogels can do to a system. So this is, you have a little bit of hydrogel, they can actually take up to 1,000 times their weight in water. So the idea is, again, they can take that water up, and you can see here, this was a high hydrogel treatment, but it actually it takes up so much water, it actually expands, and it changes the soil surface. And here you can see this is a little seedling in a hydrogel that's taken up water. So it effectively took up water, and they increased, and the Savannah soil moisture sensors found that it did increase soil volumetric water content. So pretty cool, right? So they do increase water, and these are oh gosh, these are, I'm blanking on what they're made of. So they're I will get back to you on that.
Akasha.
Yeah.
We're a little bit over time, and sorry to interrupt your talk. If you could just wrap it up, then we can have time for one question. But just wanted to say thank you so much for all that great information. But
yeah,
if we wrap it up and soon, then we can have time for a question.
Yes. I'm so sorry. I had myself until 4:15. My apologies. Yes,
no
absolutely.
Worries.
So what I will do is I will talk about, so what we found was that, yeah, thank you very much. And I'm sorry, I can't see. Something happened where I can't actually see anything. So yeah, so appreciate that. Thank you. So with Savannah's study, what we found was that it is not the end all be all. We're still doing additional studies. We found that it does improve soil moisture, but it actually hindered vegetation growth and vegetation germination. So it was the inverse of what we wanted. So I would not recommend it, but there's definitely some creative things we can do to try them. So continuing forward, we're going to continue to identify novel and creative ways to overcome barriers to restoration success. And we're thinking about those actions. And thank you. And yeah, sorry I ran over. I appreciate that. And with that, hopefully there's time for a question.
Thank you, Akasha. Awesome job. Cool stuff as always. Yeah, we have a couple minutes for question. Oh, probably one question.
I'll be quick.
Okay. How do you find out what seeds are in your seed bank? Do you bring the soil to your nursery or greenhouse and see what comes up, or are there other methods?
Yeah, absolutely. So yeah, so we often do, we collect soil in the field, bring them back to the greenhouse and do emergence trials. So we'll do these, we'll try to facilitate the germination from that. Yeah, so we usually bring them back.
Okay, I think we can squeeze one more.
Yeah.
Do you recommend setting restoration project goals to historic ecosystem reference states or anticipated ecosystem change states? If the change states are likely to be cheatgrass monocultures, is there a suite of native plant species you would recommend to outcompete annual grasses and arid ecosystems?
Oh, excellent question, Sage. And I think that one I will work to be as fast as possible. I'm happy to answer that later and kind of build on it more. But I do think thinking about, we are moving, our reference dates might be shifting a little. So thinking about what is the restoration that we're going for? What are our reference dates? I still think we probably don't want a monoculture of cheatgrass because we want to think about the function of a system. And that's kind of where we think about those abiotic function of a system. So trying to get the community trajectory on a manner is the best way possible. And I actually could teach a whole class on that. So I'm happy to add more, but I definitely don't want to keep us over. So I'm happy to, but I might stay on if you're interested in kind of that.
Yeah, and you can go back in the question and answer box and write in answers if you have a few minutes.
Yeah.
Thanks again.
Thank you. Thank you. And sorry about running over. I couldn't see you. So thank you. I appreciate it.
Thanks, Akasha. That was great. No, we're doing good. We're right on time. Sorry you couldn't see us. I don't know. There's lots of Zoom mysteries still.
Yeah, I have no idea what happened, but I appreciate it. Thank you for catching. I appreciate it.
Yeah, all good.